All right, thanks very much. We'll get started here. So today I'm going to be talking about Listeria, some of the characteristics, the contamination, and some of the patterns that you might see, and how to control it. So a lot of the topics that I'm going to cover today, it's sort of an overview of Listeria, and so each of these topics could really be their own sort of hour-long webinar. So if you want to learn more about something, a good way to do that is to sort of investigate on our website. We have the nationwide one for our integrated food safety centers of excellence, and then you can also go to the New York specific one. So we sort of cover the Northeast region and sort of try to serve as a resource to the states in our region and then nationwide as well on a few different things, one of which is environmental sampling, which sort of relates to Listeria today. So you can actually find some different webinars, other resources to learn more. I also want to let you know that we actually just developed some quick train videos. So these are short 5 to 10 minute videos that are supposed to really help you review how to sample right before you're going into sampling. So if you're ever about to sample a food facility, and let's say you're like sitting in the parking lot and you're trying to remember what you're supposed to be doing, we've got these videos available. They're not on our website yet, but they are on YouTube, so you can check that out. And we also have a Listeria cheat sheet coming out that sort of has everything you need to know related to Listeria and food. And so that'll be put on our website as well as in several different languages. So keep an eye out for that, or send us an email if you want to see that. And today's webinar is being recorded, and that will also be posted. So today I'm going to go over the Listeria basics, how to minimize Listeria contamination, and then talk about what's really required and Listeria control verification. So we're starting with Listeria basics. So Listeria is a bacterial environmental pathogen, and it's usually foodborne. So usually people who get sick from Listeria, who develop listeriosis, it's usually from a food source. It's found in the soil. It's found in the natural environment, actually. You might find it on a sidewalk or in an ATM machine, but it's found in the soil. And so that's very different from other pathogens like E. coli and Salmonella, which are primarily associated with animals and fecal contamination. They're invisible, they're small that you can't see them with the naked eye. There's no odor, so you can't really look around your facility and say, oh, that's Listeria, that's where we're going to find it. The only real method of detecting it is by microbiological testing. It's very hardy. It can grow at refrigeration temperatures, but refrigeration is actually still considered a good way to control Listeria because it does slow down the growth considerably. But Listeria is different than other foodborne pathogens in that it can still grow, even if it's slower rates, at refrigeration temperatures. So this becomes a problem if let's say you've got cantaloupe or some soft cheese, and that's got a little bit of contamination on the food, but if you put it in your fridge for a week, then that's when it starts to become a problem, because Listeria can grow in your refrigerator over that time period and become a problem, become a lethal dose. It can also survive stressful conditions, and it can persist. So Listeria is known to survive weeks, months, and even years in food facilities, continually growing and recontaminating food. It likes wet environments, so I'll talk a little bit more about that later, and it's commonly associated with ready-to-eat food. And I want to remind everyone that Listeria is heat sensitive, so if you cook your food, the Listeria will die. We sort of talk so much about Listeria that it almost feels like the superbug that can survive anything. But really, it's associated with ready-to-eat foods because ready-to-eat foods aren't cooked, so it is heat sensitive. So when I'm talking about Listeria, that's the genus. And if I'm talking about Listeria monocytogenes, that's the species. Monocytogenes is the species, and that's the pathogen of the Listeria genus. You might also hear people talk about Listeria species as a group. So that includes all of the species, for example, Listeria inocula. And so other species within this genus are not pathogenic. It's only Listeria monocytogenes that's going to make you sick. However, when you're doing testing in a food facility or in food, you might test, just in the environment, you wouldn't test food for Listeria species, but in the environment, it's more conservative to test for Listeria species because chances are, even if a non-pathogenic species of the Listeria genus can grow and survive in a place in your environment, then so can Listeria monocytogenes. So what happens if you get sick from Listeria? So most of us are healthy individuals, and so we won't suffer anything more than mild gastroenteritis. However, for susceptible individuals, such as pregnant women, immunocompromised, or elderly individuals, they might experience a more severe infection called listeriosis, and that's where Listeria gets into the bloodstream and makes you very sick. So if we compare Listeria to some other foodborne pathogens that you might be familiar with, I've got the top 5 pathogens that contribute to foodborne illness resulting in death here in the United States. Salmonella is actually number 1. It causes 378 deaths. Toxoplasma gondii, so that's the parasite that is the reason why pregnant women aren't supposed to change the litter box. And Listeria is actually number 3. It causes 255 deaths in the US each year. Norovirus comes in at number 4 and Campylobacter at number 5. Now, when we look at the top 5 pathogens that contribute to domestically acquired foodborne illness, we've got norovirus at 5.5 million, salmonella at about 1 million, and Clostridium, Campylobacter, and Staphylococcus aureus round out the top 5. But what I want to point out to you is that Listeria actually doesn't make the top 5 list for pathogens that contribute to domestically acquired foodborne illness. And that's because Listeria only causes about 1,600 cases of foodborne illness in the US each year. So what that means is that it has a very high case fatality rate, because if you think about it, it causes 1,600 illnesses, but 255 deaths, which means that if Listeria gets into your bloodstream and causes listeriosis, then you have about a 1 in 5 chance of dying. So we have to think about that in terms of outbreaks. You might have a norovirus outbreak or a salmonella outbreak that could be hundreds of cases and nobody will die. Whereas a Listeria outbreak, if you have as few as 5 cases, then you're already starting to see deaths. And that's a huge problem, not just for public health, but for food facilities as well, because that's a huge concern. So when we're talking about where Listeria is, I sort of want to zoom out for a second. This is results from a sampling project that our lab did pretty recently, where we collected soil samples across the United States from state and national parks to try to figure out where Listeria might be in the United States. And so out of 1,000 soil samples collected across the continental US, about 300 of them were positive for Listeria. And 10% were positive for Listeria monocytogenes. So I talked earlier about Listeria species versus monocytogenes. So about 10% were positive for pathogenic Listeria monocytogenes. So here we've got the distribution of Listeria by state. And I put this on here because a lot of you are calling in from Maryland. And if you look over here, we took 10 soil samples from the state of Maryland, and 7 of them were positive for Listeria. So that sort of gives you an idea of where Listeria lies in the US. And you can see some of the states have better off. Nevada, for example, Arizona. It doesn't seem like Listeria really grows that well in the environment, in those environments. But that doesn't mean that it's not going to get into their food facilities. And once it gets into a food facility, that's where it can really thrive, proliferate, and contaminate food. So even though there are some states that have less Listeria in the natural environment, it doesn't mean that those food facilities in those states are immune. But you can see here in the Northeast, we just sort of have it, we're at a disadvantage from the start. So when you're thinking about employees walking into food facilities from the street, you might want to start thinking about, what they could be tracking in. So in terms of what Listeria has looked like, or listeriosis has looked like over the last few years, I've got a graph here that shows that the incidence has actually gone down over time. So you can see here that it's actually plateaued in the last 20 years or so and sort of stuck to be the same, this orange line here. But in blue, you can see the number of solved outbreaks. So as our public health agencies get better and technologies develop, you can see that they're actually, despite there being a plateau in the incidence of Listeria, there's been a rapid increase in the number of outbreaks that they've been able to solve. The other thing that I want to highlight about this graph is that it used to be that we needed about 70 cases in order to detect an outbreak and then solve it. And now with whole genome sequencing, you really only need a couple of cases for an outbreak to even be detected. But we need to be able to solve them at that small case size as well. So what this all means is that we're going to have less epidemiological information just because we have fewer cases because we're detecting outbreaks much sooner than we used to. And as a result, that gives us a lack of statistical power. So the epi information is still critical, but it also means we need to consider all data. Does the epi data match the environmental health data, and do both of those match the lab data? So identifying a food source increasingly relies on these trace back and trace forward investigations and oftentimes environmental sampling. So the next section is about minimizing Listeria contamination. So just to make sure we're sort of all on the same page, because I'm going to be talking about whole genome sequencing in a second. So you've got your patient and they become ill. So you determine that they have listeriosis and you collect a clinical sample and you find Listeria. So those are 3 little Listeria there. So then you find out that they ate at food facility A. So you send your environmental health person in to collect some sponge samples, and you find Listeria in those sponge samples as well. So then what becomes important is that we have all those Listeria that we've collected, and we need to see if they match. So in order to do that, we get the DNA out of the Listeria, we sequence it, so similar to what you might remember from high school biology with your A's, C's, T's, and G's. You want to figure that lineup out for each of these Listeria, and then you want to match it up. And keep in mind, Listeria, the size of the genome is about 4 million nucleotides. So you're going to be matching it up across millions of nucleotide base pairs to see if these Listeria match. So I've got just a short segment as an example of the 6 Listeria that we collected in this example. And so you can see in red some of the differences. So let's say we use this first Listeria as our reference, and you can see Listeria 2 is identical, Listeria 3 is pretty closely related, 4 and 5 look a little bit more different, and 6 is identical. So let's say that the top 3 are collected from patients, and the bottom 3 are collected from a food facility. Let's say food facility A. And so what you might get from your lab personnel is you might see a phylogenetic tree. So this is sort of just like a family tree where siblings are really closely related on the family tree, whereas cousins that are more distant related or a different family altogether are going to be a little bit farther on the tree. So in this particular example, it looks like the 3 patients are pretty closely related, and they look like they're pretty closely related to the Listeria that was found in facility A on the floor. But there was other Listeria found in this facility that seems to be unrelated. So one in the drain and one at a floor wall juncture. Another way you might see this information displayed is in what's called a SNP matrix, so a single nucleotide polymorphism. And this just gives you the number of differences between the Listeria. So lower numbers mean that they're more closely related. So the other Listeria found at food facility A does not appear to be related to the first 3 patients or the isolate number 3 from food facility A. So I wanted to kind of bring you up to speed on that, because when we're talking about Listeria in a food facility, there's really 2 types. There's the transient Listeria that comes in on the product or comes in just on a person, and that's a one-time isolate, and you don't find it over time because you get rid of it during your routine cleaning and sanitation. So what we're more concerned about is, and we try to limit the amount of transient Listeria that comes into a facility. But what we're really more concerned about is something that survives over time. So it's a resident or a persistent isolate, sometimes called a PET. And what this means is that it's repeatedly found in sampling, meaning that something is going wrong with cleaning and sanitation, and it's living in what's called a niche and able to survive, proliferate, and recontaminate food over time. So what we might see is a deli case, for example, or a deli slicer. So let's give an example in context with a deli. So let's say you've got your deli area over here, your walk-in cooler back here, and maybe you have a few other sort of high-risk foods that you need to investigate. So here's an example. You've got your sites along the left side here, and then the sampling months along the top here. And so this is real life sampling data from a deli. So you can see here, NT means not tested. The little dash means that it was tested, but it was negative. And anything with a color in it means it was Listeria. And if two colors match, it means it's the same type of Listeria. So for whole genome sequencing, we can get really good at figuring out if two Listeria are the same. So for this particular deli, when we went in April, we only found 2 positives, but they were both the same type of Listeria. So one was in a floor wall junction below a 3-basin sink, and another was in the cold room on the wall. So sampling was done again in May, and you can see again we have the same site positive with the same Listeria. So it looks like either Listeria is able to survive there over time and avoid cleaning and sanitation, or it's reintroduced to that site. We also have, for the first time, a floor adjacent to the drain. In June, we have a couple of more that turn up positive, a cartwheel and a trash can. In July, you can see that, first of all, more testing was done, and it looks like this particular type of Listeria is all over the place. But for the first time, we also see a different type of Listeria in a different color. August, you can see we have 2 Listeria that we haven't seen before, this pink and this sort of tan. In September, they got lucky. We tested every site and we didn't find this blue Listeria, but we did find this green one. So some people might stop testing at this point because it looks like they solved the problem. But when we go back in October, you can see that the blue Listeria has returned, same in November and December. So you can see that these sort of random colors, that greens and pinks and tans, those are not the Listeria that we worry about. They are a problem and we do want to get rid of them, but you can see cleaning and sanitation does seem to address those particular Listeria. So what we're really concerned about in this deli is this blue Listeria, because this pattern is being found every single month except for September, excuse me. And it seems to be all over the place. So this is definitely an issue in this facility. So when I'm talking about a persistent strain, you could see in this last example that this Listeria was surviving over several months, and I wanted to remind you that it can survive over several years. So we had a deli turkey meat outbreak in 2000 where there were 29 cases, and with whole genome sequencing, they realized that there was actually an additional case in 1988 that was tied to the same food facility. So you can see over time, this Listeria was able to survive and actually cause essentially 2 different outbreaks. Another example, so this was in the US, the soft cheese, this was in Switzerland, and it was actually a seasonal cheese, and there were actually 122 cases between 1983 and 1987. So surviving over time, despite being a seasonal food and despite having the summers to shut down production and dry out a facility, it was still a problem over time. So where does Listeria hide then? If it's able to survive over time, how does it do that despite cleaning and sanitation? So I'm going to be talking about growth niches later in this presentation, but I just sort of wanted to give you an idea of an example. So here's a roller on a conveyor belt, and what happens is this conveyor belt gets cleaned during cleaning and sanitation, but this roller does not, and it's actually hollow inside. And so when you take these apart and actually look inside, you can see that they're not actually getting clean. So a big part of cleaning and sanitation should be disassembly of uncleanable equipment so that you can get inside and get the Listeria out and kill it and prevent Listeria from surviving over time. In order to test for those, you need to do sampling, and so we recommend a couple of different sampling devices. Either a lot of health departments use sponge sticks. We tend to recommend sponges with gloves. These are a little bit trickier to use because you have to use new gloves every single time, whereas a sponge stick, you don't need to wear gloves, you just need clean hands. But we recommend if you use either of these that you get the kind with the neutralizing broth so that it can neutralize any sanitizer that you might pick up with your sample. So the reason why we recommend sponges over sticks is because when you get these smaller areas, a sponge stick can't really get inside of these smaller areas, whereas a regular sponge, you can actually fold it up a little bit and get it into the smaller areas, and it has a larger surface area. Another thing to be aware of before you go out sampling is to communicate with the lab. So you need to know their capacity, how many samples they can handle. You can't just go out and take 100 samples and hope that they can process all of those. You need to know what types of samples they can handle. A lot of agencies are getting their labs to switch over to be able to handle sponges just because they're so much better at detecting Listeria and picking it up. You need to communicate the importance of using sponges. So if your lab tells you, no, absolutely not, we cannot use sponges, it's really important to communicate to them how vital they are, just because they're much more likely to, and sponge sticks too, they're much more likely to pick up Listeria than, say, Q-tip swabs, for example. And you need to communicate with the lab that the lab can have the right media on hand. So you can't just dump 100 samples on them on a Friday and hope that they have the materials they need to process them. And so this will really increase turnaround times. So basics of swabbing: you want to find places where pathogens are likely to persist. You don't want to swab flat surfaces. You want to swab 3 to 4 hours into production if possible, after the equipment has been running and shaking and things like that. Use pressure and squeeze the sponges into the right places. Make sure you look underneath things and turn things over. It's amazing how often I'll walk into a facility and there'll be a table that looks shiny and clean, and then you look underneath and it's just disgusting, as if it's never been cleaned before. You want to go until you get stopped. So if you can go underneath and behind equipment, just remember, if it's difficult to get to, it's difficult to clean. And don't be afraid to get dirty. I think I've got a picture here. So your knees should definitely look like you've been on the ground collecting samples after you've done that. And remember to dress warmly. A lot of facilities that have Listeria issues are actually refrigerated. And because of that, you want to be able to really spend time to get the swabs done and do them well. And you're not going to do that if you're really, really cold and you want to get out of there as soon as possible, especially when you go into coolers and freezers and things like that. And just a reminder, a lot of the topics I'm covering today really could be their own webinars. So if you want to learn more about this, either check our website or let me know. And this is recorded again if you want to go back to some of these lists. So when you're thinking about swabbing, there's really 3 approaches. So for food facilities, they're really going to have more of a routine environmental monitoring program. So they're really going to test all over. They're looking for Listeria because they want to actually get rid of it, but they're really also going to be making sure that they're getting a lot of samples over in the finished product and some of the drier areas of the facility, just to make sure that their control programs are working and their traffic patterns are working. For public health agencies, their approaches might be closer to an inspection or an investigation. And so their sponges are really going to be focused in the areas that are either have the highest likelihood of being contaminated and the areas that are at the highest risk of contaminating product. So when you're thinking about a surface like this, it's easy to think about swabbing like a 1 foot by 1 foot area. But we really discourage that, because when you're told to do 1 foot by 1 foot, you sort of pick a side here, and get your 1 foot by 1 foot. But what we really want is, you want the place that's most likely to be contaminated and most likely to survive cleaning and sanitation. And so in this picture on the right, that area is really the crack in between these two surfaces. So we really discourage sort of the training that tells people to do 1 foot by 1 foot, because even in this picture in the bottom right, you can see that they're getting this smooth, flat surface that's very easy to clean, when they really should be swabbing in the corners here, in this crack, and they actually should also be using a sponge and not a Q-tip. So when you're selecting sampling sites, it's helpful to know the pathogens. So for example, we're talking about Listeria. So Listeria really likes cold and wet environments, whereas if you were swabbing for Salmonella, they really like the drier environments. Although Listeria can survive in drier environments and Salmonella can survive in wet environments. So that's not a hard and fast rule. You want to identify potential niches, and remember to keep your eyes open and look everywhere, including up. So look to see the ceiling, for example. Is there condensation? Go inside of freezers, cold rooms, and remember that dirty is not always better. Sometimes you'll see something really muddy and you want to sample that, but oftentimes there's other organisms that are going to outcompete Listeria. So things that don't look that dirty but are still sort of a crack or a crevice might be your best bet. So where to sample, things like drains, floors, fatigue mats, drip pans. Again, we're recording this, so if you want to come back to these sort of examples, you definitely can. So I'll take you through a few pictures. So this is an example of a fatigue mat. For OSHA purposes, it's often nice to have these thick rubber mats, because then your employees can stand on these, and then it's going to reduce the pressure on their body for standing for long periods of time. But from a Listeria perspective, these are actually a huge issue, because Listeria actually goes and lives in the middle of these mats, and cleaning and sanitizers can't get to them. So what happens is even though you soak this whole thing in sanitizer, for example, as soon as you put it back out on the floor and you have a 185-pound person step onto it, all of the Listeria squeezes out. So we actually had a food facility where they had these mats and they were constantly coming up positive, and no matter what they tried, they couldn't seem to get them clean. So they ended up tossing out all of these mats and buying orthopedic shoes for all of their employees. And what happened was they never actually found they stopped their persistent Listeria problem by throwing out these mats. Here's an example of a floor wall juncture. You can see that this one isn't really flush with the floor, and there's actually water getting underneath here. So this is a great place for Listeria to hang out, and a really tough place for cleaning and sanitation to be effective. So this is another good place to sample. Here's an example of a squeegee. Foam squeegees, sometimes facilities like them because they're cheaper and they sometimes work a little bit better, but as a result, they're a great place for Listeria to live. So we really recommend rubber squeegees, and even those, they can become cracked and damaged, and a great place for Listeria to live. Here's an example of a food facility where they had caulking along the wall, and it wasn't done very well. So if you zoom in, you can see it's sort of rough along the edges, and another great place for Listeria to hang out. Brushes are another good example. These are actually hollow inside, and then you've got the brush bristles going inside, and it's almost impossible to effectively clean these. So these are another good example. Here's another example of brushes. This one actually has something literally growing out of it. This is a brush that's actually used for the wash process for a cheese rind. These are also, again, very difficult to clean. I think some facilities either have a replacement policy where they replace them whether they need to or not every couple of months, and some other facilities just cook their brushes as a way to kill Listeria. Anytime you have sandwich junctures, either with the floor or within equipment, that's another great example of how Listeria can get into these niches, but cleaning and sanitizers cannot. Here's an example of a drain. So this drain actually came up positive when we went to go and sample it. We actually sampled this pipe as well, and that was also positive. And maybe some of you have already noticed that there's a dead end at the end of this pipe. And dead end pipes in food facilities are really a no-go for several reasons, but basically you've got sitting water in here and who knows what else, and this is going to be a great place for Listeria to hang out. Additionally, you might see that when this drain was built, it looks like they overcut the flooring, and so you've got a nice floor crack here as well where Listeria can hang out. So all of these are great sampling sites. A lot of times we get asked, should we even sample drains? And the answer is yes. So drains are an easy way to really capture the whole room, because chances are if something's in the room, it's gonna get hit with water, and then that's gonna drain, flow into the drain. So that's a great place to start. Many facilities also have practices in place where you can, for example, high pressure hoses, and that'll allow bacteria to get from the drains onto the other pieces of equipment. So there's also the possibility that a positive in a drain will actually make it out of the drain. Here's a picture of myself and my colleague. We actually were in a fruit packing house, and we saw this drain, and when we first saw it, it actually looked like it was sealed into the floor. And when we tried to lift it together, we were actually able to lift it off. So this is us taking the cover off, and this is the inside. And so this is in a food facility, and you can see this drain is very deep, pretty old, and it's got that nice pipe sticking out. So we actually were able to take several samples in this drain, and it takes an effort. So this is a picture of me actually collecting the sponge. And so when I said earlier that you might have to get dirty in order to collect your sponge samples, I really wasn't kidding, you know. When you're collecting samples, that's not the day to be wearing a nice white coat. So I'm going to talk for a second about delis and retail. Here's that map again. So you're again, same idea. You're looking for places that are difficult to clean. So for example, deli cases, deli slicers, deli sinks, things like that are all going to be good sites to check. Cold storage rooms are another, whether you're in a food facility or you're in a retail environment, cold storage rooms, floors, shelves, and then the seals around the doors are great places to check for Listeria, and squeegees again. For retail sites that are not deli, like cheese and produce, so for example on this map there's other sections that you might want to check for Listeria. Again, you're looking at sinks, drains, floors, squeegees, the same sort of ideas and the same approach. So here's an example from a paper that came out in 2014 that shows you they collected hundreds of sponge samples in delis, and so they're number one in terms of prevalence. So they took 119 samples, and 41 of them were positive from a cold room drain. So the drain in the cold room was actually had the highest prevalence. And so you can see some other examples here along the way that would be good to check. If you, from a retail establishment, or let's say you go into retail establishments often, I would recommend you check out Haley Oliver from Purdue University. She does a lot of research related to retail, Listeria in retail environments. So I'll take you through a couple of pictures of things to look for in delis. So we already saw this one, deli cases, deli slicers. Here are some more from deli cases, and you really want to go down into the coils, into the fans, and really get to the places that aren't getting cleaned, that aren't really being disassembled appropriately. Here's a few more examples. Remember to go behind the deli counters, underneath tables, underneath sinks, behind shelves. A lot of times when you go into retail environments and restaurants, the equipment in there is actually designed to be moved around, so it's really light. And you can actually pull tables and stoves and things away from the walls, and that's great for sampling. So don't be afraid to move stuff around as long as it's safe. The other thing to keep in mind is that because they're so easily moved, they're actually oftentimes hollow. So think about hollow legs in these environments and Listeria living there, and maybe collect some samples there as well. Here's just another example of what I'm talking about there. You can see the slicer, the 3-basin sink, just a lot of different areas where Listeria could be hanging out. Here's another example of a sink. So if you want to get a sample here, you're definitely going to have to get onto the floor to actually get a good sample. And again, look at the legs here. Think about, are these hollow legs? Should we collect a sample at one of these legs? Here's a cold room. Cold rooms are another great place to sample, as I mentioned earlier, on the floors, look for condensation, and definitely get the seal around the door of these cold rooms and in the handles too. These aren't getting cleaned. More examples, I think this is a drain on the right, and you got a floor wall juncture on the left. Just other great places to sample. Deli slicers, oftentimes these aren't properly disassembled for cleaning, and so as a result, you have a lot of buildup of nasty stuff, including Listeria. So a deli slicer is actually a great example of disassembly. So when you're doing cleaning and sanitation, you should definitely be disassembling the equipment so that you can properly clean and sanitize. What this means is that to do sampling really well, it's actually helpful if you're able to disassemble a little bit. So if you're having trouble finding Listeria in an environment where you think it might be an issue, you might want to take it apart. And so oftentimes the establishment will actually do it for you because they know how to do it, but sometimes you have to do it on your own. And so you'll have to check with your local health department or local agency to see if you're even allowed to take equipment apart, because some agencies don't allow that. But if you are, and sometimes it's just helpful to know, what's involved with taking something apart. So this is my colleague Alexa, and she's taking apart this deli slicer. And you can see that there's a lot of different parts. She needs a couple of tools in order to do this appropriately. And there's, luckily we had pictures, so we knew how to put it back together. So that's also important. You don't want to put it back together incorrectly. You have to be careful too. So this, for example, is the blade of the slicer. So you want to make sure that you're being careful if that's involved. There's actually a special tool for removing this. We didn't have that at the time, so she just had to be careful. Or sometimes you have the chainmail gloves that you can use as well. Again, this is just an example of disassembly. So even though this was actually a deli slicer that was in fairly good condition, you can see that there's still some buildup going on inside the deli slicer where it's just not getting as clean, and that's a great place for Listeria. Look underneath equipment too, because you sometimes forget what could be going on underneath pieces of equipment. So you can see here, even though it looked like one piece of equipment, it's obviously made up of several pieces. So these are all things that you might want to sample during an outbreak investigation or during routine sampling. So when you're following up with a facility, a lot of times we get asked what to do if they find a Listeria in a facility, but it doesn't match the outbreak strain or doesn't match a human case. And so what we tell people is that the facility still needs to deep clean and sanitize and re-swab to confirm that they got rid of it. But it doesn't necessarily mean that you need to shut down the facility. Because most, especially delis, a lot of delis have Listeria, a lot of food facilities have Listeria. And what's important is that they get rid of the stuff that's surviving over time. So you might be more concerned, for example, if they have a lot of Listeria, even if none of it matches, or if they have Listeria in high-risk places, so like food contact surfaces, then you might want to be a little bit more proactive in your follow-up. But if they have one or two Listeria in low-risk areas, let's say a drain, and it's not matching the human case, then it's not, let's shut them down until we figure this out. It's not that. So the last section of this webinar is about Listeria control verification. So in terms of requirements, as part of FSMA preventive controls, food facilities need a monitoring program. So if contamination of a ready-to-eat food with Listeria, for example, is a hazard, then you need to do testing and environmental samples. And this applies to you if you're considered a food facility, and you're considered a food facility if you're required to register with Section 415 of the FD&C under preventive controls of FSMA as well. And if you fall under the PMO, you can actually use that. I'll talk about that in a second. So your main sort of resource for Listeria control should be the Listeria draft guidance. It is currently a draft, but the a lot of agencies are sort of using this as, you know, the standard for right now as they're getting it finalized. So you should definitely be reading this for all of your Listeria questions in terms of how to regulate Listeria. So it's only 85 pages. I promise it's a quick read. So definitely check that out. I want to highlight a couple of things that's in there. One of the things that they have is cleaning and sanitation. So when you're reviewing cleaning and sanitation SOPs, it should really have a lot of these steps. And in particular, it should really focus on the cleaning part. A lot of people like to just take their drain and dump sanitizer down that, and that's not going to clean the drain. It's actually 90% cleaning, 10% sanitizing. So you should really be using detergents and scrubbing to get surfaces clean, and then the very last step should be sanitizing. Another thing that I want to note that's in this draft guidance is they talk about keeping dry rooms as dry as possible, because water is really critical for the growth of Listeria. So if you've got a dry room, you don't want to be getting it wet, and definitely not during production. So you can really do a dry clean in those drier rooms. At minimum, for verification, you need written records. For example, you need to measure and record sanitizer concentration, and you should be doing visual inspections as well after cleaning and sanitation. It's also recommended to do ATP testing on food contact surfaces and total plate counts as a verification for cleaning and sanitation. So the draft guidance talks about the goals of environmental monitoring. And so the goals are to verify that your control programs are effective, to find Listeria and harborage sites if present, and to ensure that corrective actions have eliminated Listeria and harborage sites when in your plant. So talking about that persistent, resident, pet Listeria, all of those is referring to this third bullet. There's also a section on what your written procedures should be. So for example, they should be scientifically valid. They should specify species versus monocytogenies, identify locations, identify timing, identify the tests and the labs, and corrective actions. And for those of you who are using the PMO, which I'll mention in a second, these are actually the same. So again, this is another example of a slide that could really be its own hour-long webinar, but here's really what you need to do if you're starting a pathogen environmental monitoring program. It should really involve at least, at a minimum, all of these steps. And again, you could refer back to this in the recording. Another thing that comes up in the draft guidance is that 5 sites for sampling should be food contact surfaces, and 5 sites should be non-food contact surfaces. So I caution you to be careful with this one, because if you do end up doing 5 sites for food contact surface, I want to make sure that you're aware of the things that you need to do in terms of corrective actions for a positive. You have to be aware of the consequences of a positive. Don't just test the sites and then ignore it if it's a positive. So if you have a food contact surface positive, you have to have a plan in place of what you're going to do. You obviously need to retest, possibly hold or recall product until you find out what's going on. So here's a little flow chart that's actually present in the draft guidance. For low-risk food, when I visit food facilities that are really good on food safety, if they're a low-risk food, they actually don't do a whole lot of food contact surfaces, and they do much more zones 2 and 3, so the surfaces next to the food contact surfaces. But if you're a higher risk food, for example, soft cheeses, for example, you should do food contact surface testing, and it should be negative. But again, have a plan in place so that you're not just testing and hoping that it's negative every time. I keep mentioning the PMO. If you're covered under the PMO, then you should be covered in terms of Listeria. You just need to check page 396, Appendix T, preventive controls for what you're supposed to do with regards to Listeria environmental monitoring. Finally, I want to talk to you about how you might check to see if your sampling plan is working. So here's an example of research from 7 different cheese plants where we did routine sampling. And so you can see the prevalence from several months of sampling. And so if I were to just look at this based on the prevalence, I think I would want to eat at facility F or facility I, because, or even facility J, because they look like they've got the lowest prevalence. And I think I would be most worried about eating cheese from facility H, G, and E, for example. But when we sent in, so all of this testing was done by the food facility themselves. And so when we sent in outside experts to pick sampling sites and to actually do the sampling, sites from zones 2 and 4 over 6 months, we actually found out that the prevalence was quite different from what they were finding in the routine. So facility F, out of 334 samples, they didn't find anything when they were doing the testing, but when our outside experts came in, they found 6% prevalence. Same with J, there was also a huge jump. So this is a great way to validate whether or not your routine sampling is looking in the right places and is actually doing the swabbing correctly. So you want to be a facility that's green, where someone from the outside actually has a harder time finding Listeria, because really the people who are doing the sampling in the facility itself that are familiar with the facility should be better at detecting it and finding it than someone coming in from the outside that's never seen the facility before. So to summarize, Listeria can contaminate ready-to-eat foods, causing illness and death. Verification and validation of cleaning and sanitation programs is essential, as Listeria can survive in a food facility over time in those niches where cleaning and sanitizing is ineffective. And lastly, most ready-to-eat food facilities are required to have a scientifically valid sampling program. So that's all I've got, so maybe we can open it up for some questions. Let's see what we got.
Hey, Genevieve.
Yes.
Hi, this is Renato. We have questions in the Q&A,
Okay.
and we also have a question in the chat. Do you want me to read, or you want
Yes, that'd be great.
Okay, so in the Q&A, we have a comment from Bill. He says, the sampling sponges shown are fibrous cellular material, and the broth mentioned is DE broth. And then he has a question. What are your thoughts about high-capacity neutralizing broth and polyurethane sponges versus the above mentioned?
So I'm less familiar with the other type of neutralizing broth and polyurethane sponges. The ones that I mentioned are the ones that we typically use. They seem to work really well for us. So if you're considering something else, either because you think it works better, or because it's just a better price, or because your lab is better equipped to handle it, one of the things that you can try is using both in food facility and seeing which seems to detect Listeria better, and then just choosing whichever one works better for you. But as long as it has that neutralizing broth to neutralize sanitizers, and it has the surface area that's going to help you pick up Listeria if present, then you should be okay.
Great. So the other question was in the chat. It's from Alicia Johnson. She says, I was told that Listeria monocytogenes cannot grow in food with a pH of 5.0 at 41 Fahrenheit. Is that true? I thought it was able to grow down to a pH of 4.4 at a temperature of 41 Fahrenheit.
Yeah, so it can. So if you check, I think it's the FDA. I think those are the right numbers, 4.4 and 41. I would have to check to be sure. I don't have them in front of me. But yeah, Listeria can grow in some pretty extreme conditions. And that's not to say it grows well, for example, at pH 4.4. That's really the limit. But it can grow, and it can definitely survive. So you can use acid as a way to control, but it has to be pretty extreme. Hopefully that answered that question.
We have another question that pop up right now. Are you aware of Listeria associated with cut vegetables?
So I am aware of Listeria in facilities with cut vegetables. I'm trying to think, there's been several produce outbreaks, and I'm trying to think of one that's cut vegetables. I'm guessing you mean besides lettuce. I guess lettuce would be a good example of Listeria with cut vegetables. Yeah, does that answer that question, Listeria? Yeah, oh, besides lettuce, okay. Yeah, I can't think of any off the top of my head of cut vegetables, but it's definitely in those operations. I do know that 100%. And just because there hasn't been an outbreak detected yet doesn't mean there hasn't been an outbreak with cut vegetables and Listeria.
Okay, yeah, there is a comment now. There have been Listeria monocytogenes in frozen vegetables.
Right, yes, frozen vegetables. And that's definitely an issue when we're talking about smoothies, for example. So a lot of cut vegetables are being used in ways that people didn't originally think that they were going to be used. And smoothies is a great example of that. People love throwing chopped vegetables in their smoothies in the morning, and then they have them sitting in their car on their desk all day, and Listeria can grow and definitely become a problem. So yes, thank you to whoever sent that in.
Right, that was then, and she also asked, what about presence of multiple species of Listeria in a firm? How is that associated with the presence of Listeria monocytogenes in a firm?
Yes, so we sort of saw this with that deli example where we had the blue type of Listeria, and so that was the one that was the major issue because we kept finding it. But there's definitely a chance you're going to find not only multiple types of Listeria species, but also multiple types of Listeria monocytogenes. So that's definitely something to be aware of. Even within one sampling site and one sponge, you might find multiple types of Listeria. So that's definitely an issue as well. So what we found is that you might even, if you're looking for an outbreak strain and you're not finding it, you may even need to select multiple colonies for whole genome sequencing in order to find the one that you're looking for. Just because multiple types of Listeria can live together. In terms of your response to those, if a food facility has multiple types of Listeria, you should be more worried about the overall prevalence. Are you finding it in a lot of different spots? Are you finding it in high risk areas, for example, food contact surfaces? And if so, that's when maybe an outside cleaning and sanitation team should be brought in to do a deep clean of the place, and maybe cleaning and sanitation SOPs should be reviewed. But if you're finding like one or maybe even two Listeria in a food facility, that's not uncommon. So that shouldn't be, reason to come down hard on that facility, for example.
So following up on that, Genevieve, another question, what's your advice? Oh, well, it's not a question. What's your advice for a facility that has found Listeria over a longer period of time?
Right. So it again kind of goes back to the prevalence and where they're finding it. So if they're continually finding it in, for example, a sandwich juncture on a piece of equipment, and they keep finding Listeria, even if they're not sequencing it, they don't know what type of Listeria it is. That's a case where they either need to review cleaning and sanitation, where they need to disassemble that during cleaning and sanitation, or they need to get rid of that piece of equipment. Obviously, that's not a perfect solution, but if it's a place where Listeria is continually able to survive and be detected, then that's an issue, and they need to address that, no matter what the sequencing type of Listeria. If you're in a facility where you're finding different types of Listeria every single time you go, and it's more than one or two, then cleaning and sanitation procedures again need to be reviewed. So yes, I guess it's on a case-to-case basis, but if they're continually having Listeria found in a food facility and high numbers of it, then yes, you need to review that those procedures. Hopefully that addressed that. Feel free to send a follow-up question if I didn't quite answer your question.
I think it did. We don't have any other question right now, but I would like to remind everyone that we have a survey. I posted a link, and I'm actually going to post right now again in the chat box. So if you can go to the link and complete the survey, that would be great. Let me see if there is any other question. Okay, it looks like we have. How can you verify supply if it is associated with a product that is brought into the facility?
Okay, so that's a good question. So we have seen cases where you'll find Listeria from two different facilities that actually appear related. And so oftentimes that's caused because they have a common supplier. So what you need to do is you need to go a step further and figure out where that supply is coming from. Is it being contaminated on a common truck? Is it coming from the same field? If it's produce, is it coming from a previous storage facility or otherwise just a previous facility where a food is coming from? And so you sort of had to follow the supply chain to the previous location and do sampling there and sort of investigate and do an inspection or investigation. So that's actually a great question because you really do have to keep that in mind. You can't just assume that because you find the matching Listeria to a case that it's because it's from that facility itself. It actually could be coming in on product from another location. Hopefully that answered that question. But in order the facility, you should be able to ask them, where is this coming from? If they can't answer that, then that's a problem.
Thanks. Let's see. I think there is another message. Okay, so I think we don't have any more questions then.
Okay.
Okay, thank you. Thanks, Renato. Thank you so much, Genevieve. That was really excellent, and I really appreciate your time and your expertise and for being willing to collaborate with us on this project. Again, I wanted to recommend that people fill out the survey because that'll help us to understand, whether or not you found this useful, and if you have any comments about how to make it better, we would appreciate that as well. Additionally, if I did send you a sign-in sheet to have you sign in, if you were hosting the webinar, with a large group, it'd be nice for us to know exactly who in your group attended, and we'd also like to have a list of the attendees so that we can send out any further information that we may have. So anyway, I'll end the meeting now, and thank you again, Genevieve, and thank you, Cornell, and thank you, Renato.